Hello, everyone, and welcome to today's The Scientist webinar. I'm Betsy Young, Director of Creative Services for The Scientist, and I'll be moderating our discussion. Today, our speaker, Dr. Steve Horvath, will be discussing how the epigenome influences aging and the approaches scientists are taking to study the possibility of targeting epigenetic modifications for therapeutic intervention. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speaker will address these during the Q&A session following the presentation. To ask a question, simply click on the Ask a Question tab and type your query into the question box located on the bottom left of your screen. We'll try to address as many of these questions as we can during our Q&A session. Our webinar platform is user-friendly. You can expand the presentation window by simply clicking on the diametrically opposed arrows in the upper right-hand corner. This will maximize the display within your screen. The webinar will be archived on the Scientist website, and we'll send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors. Zymo Research has been proudly serving the scientific community for nearly 25 years by providing innovative, reliable, and high-quality research tools, especially in the fields of DNA methylation, epigenetics, and aging research. Zymo currently offers a high-throughput biological age prediction test based on the analysis of DNA methylation patterns at hundreds of highly informative gene loci. Find out how to accurately quantify the aging process in both human and mouse research samples by visiting their website at www.zymoresearch.com/DNAge or by email at services@zymoresearch.com. Active Motif is the industry leader in developing and delivering innovative tools to enable epigenetics and gene regulation research. We are committed to providing the highest quality products and superior service and support to serve the life science, clinical, and pharmaceutical drug discovery communities. Whether you're an expert in the field of epigenetics or a researcher interested in integrating epigenetics research into your studies, Active Motif offers a comprehensive portfolio of epigenetics research-related products and services and the support of our team of epigenetic experts to provide complete and innovative solutions to tackle your scientific inquiries. Usurance, powered by Life Epigenetics, is bringing step-change technology to the life insurance industry. Usurance is applying the most advanced methods in predicting human longevity based on the science discovered by Dr. Horvath, featured in today's webinar. To find out more about how Usurance is changing the life insurance industry, go to usurance.com. Our sponsors have provided us with some helpful resources related to the role of epigenetic modifications in biological age and the aging process, and we've posted these in our resource list located on the left of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker, Steve Horvath. Dr. Horvath is a professor of human genetics and biostatistics at the David Geffen School of Medicine at the University of California. He received a PhD in mathematics from the University of North Carolina at Chapel Hill in 1995 and a doctorate of science in biostatistics from the Harvard School of Public Health in 2000. Dr. Horvath's research lies at the intersection of aging research, epidemiology, chronic diseases, epigenetics, genetics, and systems biology. He works on all aspects of biomarker development with a particular focus on genomic biomarkers of aging. He developed a highly accurate multi-tissue biomarker of aging known as the epigenetic clock. Dr. Horvath developed systems biologic approaches such as weighted gene co-expression network analysis, which lend themselves for integrating gene genomic datasets. These methods have been used for a broad spectrum of age-related diseases, including neurodegenerative diseases, cancer, and cardiovascular disease. Dr. Horvath?
Yes. Hello, everyone. Thank you so much for your interest in this topic on DNA methylation biomarkers of aging and development. And on my first slide, I show you the medieval view of mortality. There's an hourglass. Now in the 21st century, we have come to an understanding that there is, in fact, an hourglass of mortality, but it is really the DNA molecule. And so I really like the right panel that shows, on the one hand, the DNA as the carrier of the genetic information, but also as a gigantic clock, which allows one to estimate the age of really any cell type that contains DNA. So I want to start with the motivation. Here you see a bodybuilder, a 71-year-old man, and according to most clinical biomarkers, such as body mass index or grip strength, he probably looks very young, like a young man. However, according to molecular biomarkers, he's probably age appropriate, around 71 years old. And so the message here is that molecular markers will not be misled by outer appearances. And the goal of my research and that of many others is to develop these molecular markers that measure aging in its various manifestations. So this task is actually quite difficult, as you can see from the conceptual framework. So the molecular biomarkers in the middle of the graph relate certainly or hopefully to an innate aging process in the upper panel, but at the same time, they undoubtedly also relate to various stress factors. Could be smoking or metabolic stress or many other confounders. And has made it very difficult to develop molecular biomarkers of aging. Now, one thing though is for certain, and this is that whatever molecular biomarker one develops for aging, it really should relate strongly to chronological age. And on the right-hand side of the chart, you see age-related phenotypes. We're talking here about various diseases and even mortality. Now, that is undoubtedly driven by innate aging processes, but also, of course, mortality is very much influenced by various toxins and stress factors. And so it's actually very difficult to distinguish age-related changes due to innate aging processes from those that come from confounders. Now, in my presentation, I will focus on molecular biomarkers that are based on DNA methylation. Why? Because I think they work very well. So here I show you on the next slide something known as cytosine methylation. So the DNA molecule has, of course, 4 nucleotides, and one of these, the C, cytosine, can occasionally be methylated. And methylation has been studied for decades, and it's well understood that methylation plays a crucial role in development and cell differentiation. But of course, it's increasingly being recognized that DNA methylation also plays an important role in aging. And on the right-hand panel, I show you really the applicability of the epigenetic clock across the entire lifespan. As I will show to you later, the epigenetic clock applies to prenatal samples, to childhood, middle age, and certainly also old age. The epigenetic clock method turns out to be highly platform independent. However, in my work, I mostly focus on DNA methylation data that were generated on the Illumina methylation platform. And the latest platform is called the Epic chip, which measures 850,000 locations on the DNA, but older versions are the 450K array and the 27K array. And the pan-tissue epigenetic clock that I will describe shortly applies really to all of these platforms. Now, what such a chip measures are methylation levels at a CpG location. And so in your data, for each CpG, you get a number between 0 and 1, which indicates in certain ways the proportions of chromosomes that are methylated at a particular locus. So for example, if you have a blood sample comprised of 100,000 blood cells, and if about half of the chromosomes are methylated at a particular locus, then you get a number of 0.5. Now, the pan-tissue epigenetic clock, which is also known as the multi-tissue epigenetic clock, is based on an algorithm that can be carried out in 3 steps. So in step 1, one needs to measure the DNA methylation levels of 353 CpGs. And you can, in principle, use any method of measuring these methylation levels. Next, you form in step 2 a weighted average of these 353 numbers. Remember, each number takes on a value between 0 and 1, and you form a weighted average, and that's not yet in units of years. So in step 3, we transform this weighted average, and then it's in units of years, and the result is an age estimate that is then known as epigenetic age, or more precisely as DNA methylation age. The stunning property of this algorithm is that it's the same definition for every tissue and cell type. It's always the same way of forming a weighted average. And so this human pan-tissue age estimator applies to all tissues and cell types and fluids that contain DNA, with the exception of sperm. And the right panel shows you the accuracy of this method in test data. And you see a correlation as high as 0.96. The correlation is so high because we consider here samples from age 0 to 100 or so. So if you have a broad age spectrum, you see very high correlations. One question is, how does methylation age change across time? And here I show you some spaghetti plots that show on the y-axis the methylation age, on the x-axis chronologic age, and 2 points are connected if they came from the same person. And what you see on the left panel from the in-Chianti cohort is that an individual who had a certain, was above the red line, in certain ways was older than expected at a young age, this same person tended to be older also at a later age. So it turns out if your epigenetic clock ticks faster early on, it will also tick faster later on. So there's the insight here is that epigenetic aging is to a considerable extent under genetic control. On the right panel, you see occasionally a person whose age estimate was reversed, right? But I think this is due to measurements errors or technical artifacts. I want to say for 98% of all individuals, methylation age truly increases with time. So if you measure somebody's age at age 40, and then let's say methylation age is 45, then you measure it 10 years later, then the methylation age estimate will be 55. You know, it's truly measuring the passage of time. Now, I've been struggling with a good term. How does one call a biomarker of aging and development that truly applies to the entire life course from cradle to grave? And one way to call it is a life clock, but I actually prefer the German name Lebensuhr. So although this talk is mainly focused on age-related diseases, I do want to emphasize these tools very much apply to the entire life spectrum. And to show you that, I show you here the multi-tissue or pan-tissue methylation age estimator in training and test data. And you see that early on at age 0, from age 0 to age 20, there's a fast rate of change, but it still tracks aging. But after age 20, the change is almost constant. And so this, by the way, for the experts, the y-axis shows the results from step 2 of the epigenetic clock algorithm that I just presented before I calibrated it. The red line is the calibration curves that I use to map the methylation age estimate to chronologic age. So this insight that these biomarkers apply to the entire age spectrum really supports an epigenetic clock theory of aging, which is to say that this DNA methylation age is a continuous readout that links biological processes that play a role in development and later during adulthood in tissue maintenance, but ultimately they play a role in organ decline and aging. So we interpret this as an example of antagonistic pleiotropy, where purposeful molecular processes that play a role in development and tissue maintenance ultimately have adverse consequences later in life. Now, these kinds of theories have been presented in various ways or incarnations, but what is new about this particular clock theory is that this emphasis on the fact that the methylation age is really the biomarker that tracks these purposeful and then ultimately adverse biological processes. I also want to highlight that I don't think that epigenetic changes are the only reason why we age. As you know, there are many hallmarks of aging, but we certainly know that it's a contributor to aging. Now, one drawback of these molecular biomarkers is that their precise mechanism is not yet understood. So we believe that these age-related methylation changes lead to a loss of cellular identity and also lead to a decline of fully functioning somatic stem cells. I want to present a new epigenetic clock for skin and blood samples. We just published it today. Let me explain why we looked at it. So the original pan-tissue age estimator actually led to fairly large errors in fibroblasts and also in other skin samples. And that's kind of frustrating for people who like cell culture studies because many people work with fibroblasts. And so there was really a need to develop a new and more accurate epigenetic clock for fibroblasts, but we also developed the same biomarker for keratinocytes and endothelial cells and many other cells that are often used in the dish. And here I show you a comparison of the new clock, the skin and blood clock in the left panel versus the original pan-tissue clock. And I show you data, and these are now test data. The upper panels are in fibroblasts, lower is buccal epithelial cells, keratinocytes, and dermis and epidermis samples. And you see for all of these 4 data sets, for each of these 4 data sets, the new age estimator is much more precise. The median error is better. but also the correlation coefficient is higher. Now, the skin and blood clock also is called, among other things, the blood clock, right? Why? Because it actually also has superior accuracy in blood samples. And to show you that, see the upper panel, which is the new estimator versus the pan tissue estimator in the bottom. I mean, the correlations are similar, but the error is a little bit better. So this new clock has certainly a forensic application. So you can apply it to buccal cells, saliva cells, blood cells, and to accurately measure age, but you may also apply it to dating, where people often lie about their age. I want to come now to a serious application, the Hutchinson-Gilford progeria, which is due to mutations in the LMNA gene locus, and different mutations lead to different types of the disease. Some are more severe than others. The classic Hutchinson-Gilford progeria is a childhood progeria, and many individuals don't live more than 20 years, unfortunately. Now, we analyzed fibroblast samples from the Progeria Research Foundation and applied then this new skin and blood clock to them. The upper panel shows you how chronologic age at the time of the collection of the fibroblasts relates to the estimated age, and the dots are colored by disease status. Red dots are classical progeria, green dots are non-classical progeria. And so if you look at panel B, it's the same analysis, but attention is restricted to individuals who are less than 10 years old. And overall, you see that diseased individuals exhibit a methylation age that's a little bit older than that of controls, the black dots. And a formal statistical analysis bears that out. So now that's a nice finding in certain ways, because it shows that the new skin and blood clock shows the expected result that progeria is associated with older epigenetic age. But I do want to mention that the original pan tissue clock did not find that result. So in certain ways that supports the view that for fibroblasts, maybe the original clock is not ideal. On the other hand, it also shows that this result is perhaps not robust with respect to what biomarker is used, which kind of methylation biomarker is used. I briefly mentioned that I've also analyzed other types of progeria, so-called Werner syndrome, also known as adult progeria. And there we do find that Werner syndrome individuals have older blood than controls, according to the epigenetic clock. And finally, I want to briefly mention that Down syndrome, which many people consider as a segmental progeria as well, Down syndrome actually has the strongest effect by far. Down syndrome individuals have the oldest methylation ages, no matter how you measure it. Moving to the next slide, I briefly want to mention that the skin and blood clock does track what is known as cell population doubling levels, or passage number. And so in other words, if you split the cells in a dish, you actually increase their methylation age. But even after we adjust for population doubling levels, this result for progeria holds up. If anything, it becomes stronger. So as I mentioned, the skin and blood clock outperforms an existing biomarkers with respect to age estimation in skin and blood cells. It's based on 391 CPGs. It's really tailor-made for the cells I mentioned. But interestingly, it also applies to some extent to cell types that weren't used in the construction. So, for example, neurons, glia, or various brain tissue samples, liver samples, and even bone samples. So it turns out many of these age estimators lead to fairly high age correlations also in other tissues. Now, the limitation of the skin and blood clock is that it's actually not the best predictor for lifespan, based on blood methylation data at least. So I will briefly show you that many of these methylation biomarkers predict lifespan and morbidity. Certainly the skin and blood clock does that as well, but we have better predictors of lifespan. So in general, there's no longer any doubt that these methylation-based biomarkers predict time to death. Why do we know it? Because there are many independent groups who have looked at that question using large cohort studies, and they all had roughly the following design. People collected blood samples in the 1990s, put them in the freezer, and after 30 years or so, they went back to these blood samples and generated methylation profiles. And therefore, one can see whether the methylation data from 30 years ago would have predicted lifespan. Because, of course, for these individuals in these cohort studies, we also knew whether they were still alive or whether how long they lived. I want to describe a novel predictor of lifespan, DNA methylation pheno-age or phenotypic age, which is based on 513 CpGs. I mention it because it's a better predictor of lifespan than the other biomarkers I just mentioned. And it predicts all-cause mortality. It predicts also cause-specific mortality. It relates to coexisting disease count, the multimorbidity indices, and various measures of physical functioning. And it relates to a host of other conditions, age-related conditions. Now, I want to give you a bit of a sense how good such a lifespan predictor is. So here we show you two Kaplan-Meier curves that show the proportion of individuals who are still alive versus time or chronologic age. And the blue curve depicts the survivorship for individuals who are 10 years younger than expected according to this pheno-age estimator, whereas the green curve shows you the other end of the spectrum. So these are individuals who are 10 years older than expected. And you do see a fairly good separation, you know. Having said this, well, it's in the eye of the beholder. Some people will think of it as a huge separation. Others will say, well, it's not such a big separation. For one, it's statistically hugely significant, you know. So there's no question. Now, the pheno-age is associated not just with all-cause mortality, but also time to coronary heart disease, or conversely, time of health span, or the decrease in likelihood of being disease-free, and so on. Now, I do believe that epigenetic clocks are tremendously useful for identifying and validating anti-aging targets in clinical trials, but I do want to present a reality check, which is that epigenetic clocks do not stand out in terms of lifespan prediction per se. Many alternatives based on blood pressure, smoking levels, frailty indices, lipid levels, glucose levels, all of these biomarkers that a doctor measures in a clinical setting, they are of course hugely informative as well. And these epigenetic clocks will not replace these standard biomarkers from the clinic, but they do enhance them. So there's no doubt that these epigenetic biomarkers add to the predictive accuracy of lifespan predictors. They're in certain ways, to some extent, independent of them. The multivariate models where you include all of these clinical biomarkers and on top of it you include a methylation-based biomarker would leave the methylation-based biomarker as a significant covariate. The great advantage of these epigenetic clocks, in contrast to many other clinical biomarkers such as glucose levels, is that they must be proximal to an innate aging process. Conversely, think of glucose levels. They could very much be elevated, greatly elevated, in an adolescent who is severely obese and developed early onset diabetes. And then the question is really obesity and elevated glucose levels truly a root cause of aging? It's a matter of debate. They might be. Now, conversely, with these methylation age estimators, there is really no debate. They must relate to a root cause of aging because they relate to the properties of the fundamental molecule of aging and development, and our longitudinal studies also show it. The other great advantage of epigenetic clocks is that they can be applied to cells in a dish. You can do these in vitro studies. And of course, you can use them as phenotypes in genetic studies for finding, for example, genes that modulate the intrinsic aging rate. I want to describe results for various anti-aging treatments. In certain ways, what can we do against epigenetic aging? So the ultimate approach for resetting the epigenetic clock is to apply Yamanaka factors. That completely resets the epigenetic age of a fibroblast to that of the prenatal cell, for example. Beyond that, hematopoietic stem cell transplantation resets the epigenetic age of blood of the recipient to that of the donor. Now, to explain that finding to you, assume there's a 50-year-old man who has a severe form of leukemia, and so he needs a bone marrow transplant. After his blood is completely removed through radiation and other forms, and he gets now a transplant, a bone marrow transplant from a 20-year-old donor, you may wonder, well, what will be the resulting age of his blood after the blood is reconstituted following that transplantation? And as I mentioned, it's actually completely the age of the donor. And that finding actually holds also in the long term. If we followed this recipient of the bone marrow transplant for 20 years and asked in 20 years what's the age of his blood, it would still be reflective of the chronologic age of the donor. So the stem cell niche, surprisingly, does not seem to affect the methylation age. However, there are several caveats. The first thing is that graft versus host disease does increase epigenetic age of blood, unfortunately. And of course, a bone marrow transplant is a rather risky and dangerous procedure. So this is not a viable anti-aging intervention, at least at this point. I want to mention another intervention, hormone therapy. We never had a chance to look at testosterone treatment in men. However, we had a chance to study menopausal hormone therapy in women. And interestingly, hormone therapy kept the buccal epithelial cells young, and you see that on the right-hand side. However, it did not rejuvenate, this therapy did not rejuvenate the methylation age of the blood samples of these women. And this result makes sense in certain ways because buccal cells have more estrogen receptors, so they are actually more susceptible to these hormones. By contrast, blood cells have far fewer estrogen receptors, so they might not be able to pick up this beneficial effect. Now, as many of you know, menopausal hormone therapy also has risks. It's by now a controversial therapy, and so I'll leave it at that. What about more conventional interventions that everyone thinks about, such as healthy diet, physical exercise, and education, and so on? So we looked at it in almost 4,500 individuals, mainly from the Women's Health Initiative, and we analyzed these blood methylation data with respect to two different epigenetic clocks. The extrinsic epigenetic clock, which is an enhanced version of the Hannum clock from 2013, and also the intrinsic epigenetic aging clock, which is really the pan-tissue estimator, but now adjusted for differences in blood cell counts. So in certain ways, the right-hand side shows you results that are independent of blood cell counts, whereas the left-hand side shows you results that are actually confounded by changes in blood cell composition. And so what did we find? So in the middle, you see all the factors that affected both clocks. Metabolic syndrome, higher glucose levels, obesity, and so on. All of these factors were associated with faster epigenetic aging. The left-hand side shows you factors that affected the extrinsic clock. So everything you know from your parents about eating healthy fruits and vegetables, eating fish, higher education, doing exercise, and also moderate alcohol consumption, all of that is good for you. Why? Probably because it reduces inflammation in some shape or form. By contrast, the intrinsic epigenetic clock is much less susceptible to these lifestyle factors, and it's to a greater extent under genetic control. We actually did find that poultry consumption had a weak effect, a beneficial effect on the intrinsic clock, but that could easily be a false positive finding. So, now here I show you the actual data. So when you carefully look, there are correlation coefficients called BICOR and p-values. And what you notice is that these correlation coefficients, more often than not, are very weak. Look at the bottom, where you see the effects of alcohol, also of smoking and exercise, income, education. So education actually is associated with a lower epigenetic age according to the extrinsic measure, which is confounded by blood cell composition. But notice the correlation coefficient is only minus 0.1. That's a very weak correlation. The p-value is so significant, namely 10 to the minus 10, because we studied here over 4,000 individuals. So overall, many of these lifestyle factors have only a weak effect on epigenetic aging rates. I want to draw your attention to these nutrient measures, mean carotenoid levels, which measure in some shape or form vegetable consumption, were also very much associated with slower epigenetic age according to the extrinsic measure, p-value 10 to the minus 9. So yes, eating vegetables is good for you, but again, the correlation is only minus 0.13. That's a weak effect. Right above it, you see various measures of dietary variables, but these are self-reported measures. And what you notice, self-reported vegetable consumption was actually not associated with any of the measures. Why is that? Well, because that's recall bias. When people fill out these dietary forms, they know they should be eating broccoli, so they might tick it off as that they ate it, but in reality they didn't. Whereas these blood-based measures of vegetable consumption, these, for example, mean carotenoid levels, they cannot be fooled, and they detect who benefits from these from the vegetable consumption. So I want to give you a status overview of these methylation-based biomarkers. So in 2013, we realized that we, meaning we, the research community, realized that these methylation levels allow one to very accurately measure age. By 2015, there were several papers that show that the discrepancy between methylation age and chronologic age, this delta age, actually predicts mortality. It relates to cognitive functioning, obesity, Down syndrome, and so on, many age-related conditions. Now, in the year 2018, we have very powerful predictors of health span and lifespan. We have custom clocks for these ex vivo studies, and several labs have published mouse epigenetic clocks. Now, in the year 2021, 3 years down the road, I expect that we will have biomarkers of aging in many different species, certainly in all mammals. So in my lab, we work right now on these methylation studies in 50 different mammals, and we want to develop really these biomarkers across mammals and within mammals. Now, as I mentioned earlier, one of the great challenges of these biomarkers is that we don't completely understand the molecular causes and consequences of epigenetic clocks. It's a bit difficult. So some clocks, for example, the pheno-age clock, actually does capture aspects of immunosenescence and inflammation and also age-related changes in blood cell composition. And it also relates weakly to telomere length. However, the multi-tissue clock and even the skin clock, they don't exhibit that type of enrichment for pathways. They probably relate to properties of stem cells or hematopoietic stem cells in blood and so on. One of the reasons why it's so hard to come up with a biologic interpretation of these biomarkers is that they really capture aging so well. And there's really not a single cause of aging. So it's no surprise that these biomarkers actually relate to several properties of the aging process. I want to mention that an epigenetic clock is not a marker of cellular senescence, and there are several reasons for it. One reason is that the methylation age actually does not, you cannot arrest it if you immortalize a cell, for example, by hTERT expression. So these are fibroblast studies from Ken Raj. And here he showed how cumulative population doubling level on the left-hand side, on the x-axis, relates to methylation age. And you see findings for an immortalized cell versus a non-immortalized cell, and you really see that immortalization doesn't stop the epigenetic clock. Ken has done some additional studies where he looked at different ways of inducing cellular senescence, or replicative. So replicative senescence is associated with an increase in methylation age in these endothelial cells. And also on the right bars show how oncogene-induced senescence also induce an increase in methylation age in these endothelial cells. However, surprisingly, radiation-induced senescence does not seem to increase the methylation age. So the finding, the results of these studies is really that different avenues for inducing senescence can be distinguished by their effect on the epigenetic clock. I want to move to another topic, telomere length. So the upper panel shows you how leukocyte telomere length correlates with chronologic age in the Framingham Heart Study. So each dot is a person, and you see a correlation of minus 0.27. That's the famous statement that telomeres shorten with age. The panel underneath shows you how methylation age relates to chronologic age in the very same individuals. And you see, of course, a much stronger correlation, 0.81. Now, this is not a cherry-picked study. Anybody who has ever looked at it has found the same thing, that yes, these methylation age biomarkers have a much stronger correlation with chronologic age. And several papers have explored the relationship between epigenetic age and telomere length, and actually both of them seem to be independent predictors of mortality. But then if you correlate them, these two these markers, for example, the multi-tissue or pan-tissue methylation age biomarker with telomere length, after you adjust for chronologic age, there's really no significant correlation. And we know it because we've looked at over 10,000 individuals and from many large-scale cohorts. In most situations, the correlation was statistically not significant. And in principle, that would then lead to the conclusion that telomere length is just a different biomarker from these methylation changes. There is no connection. However, Akelu has carried out a large-scale genome-wide association study of epigenetic aging rates, and the finding for the pan-tissue biomarker, the intrinsic epigenetic age acceleration, which is importantly not related to changes in blood cell types, completely independent of blood cell composition. So after she uses that as a quantitative trait in a GWAS genetic study, she found that one of the top hits was the TERT locus. Now, TERT is an important subunit of telomerase, which maintains telomere length. So that finding was completely surprising to me in light of the previous slide. And the right panel shows you how the p-values for the intrinsic epigenetic age acceleration change across the TERT locus, and the blue dots show you the p-values for leukocyte telomere length, and you see perfect overlap. So that's stunning. Now, these GWAS studies also implicated, to a lesser extent, other genes that play a role in telomere maintenance. So you see that p-value for TERT was 10 to the minus 12, but for the other genes it's much less, it's 10 to the minus 4 and so. But overall, there was this consistent association between telomere maintenance genes and genes that relate to epigenetic aging in blood. However, when we did a more careful analysis, it turned out that these SNPs in the TERT locus had a pleiotropic effect on telomere length and intrinsic epigenetic aging. As a matter of fact, there was a paradoxical finding. SNPs that in the TERT locus that were associated with longer telomeres were actually associated with lower intrinsic epigenetic age acceleration. It's paradoxical because what it indicates is that if you win the genetic lottery by inheriting certain genetic variants in the TERT locus that give you longer telomeres, then you actually lost the genetic lottery with respect to the epigenetic clock, because these variants actually were associated with faster ticking of the epigenetic clock. So it's a paradoxical association. Now, this paradoxical finding is supported by an astronomically significant meta-analysis p-value. We already validated it in multiple large cohort studies since the publication. We also could validate it in different racial groups. We had in vitro studies that supported it. So this association, this paradoxical association, is irrefutable. But the biological interpretation is less clear. And so here on the next slide, I show you different explanations and different interpretations. And really, future research has to tease that apart, which of these interpretations is applicable. But one interpretation would say that, well, if TERT can be interpreted as an oncogene, then this finding mirrors the finding that epigenetic age acceleration accompanies oncogene induction. But another interpretation of our finding is that hematopoietic stem cells that have longer telomeres, that maintain their telomeres for longer, they are around for longer, they live longer, and therefore their methylation age will also increase. There's another more speculative interpretation, which is that TERT expression might allow cells to record their proliferation history. In certain ways, an epigenetic clock might be a mitotic clock in certain circumstances. And there's some evidence for that. I do want to emphasize that in general, for most cells and for tissues, the methylation clock, the epigenetic clock, is not a mitotic clock. It doesn't measure the number of cell divisions, but in certain circumstances it might be. Interpretation 4 is that epigenomic maintenance might relate to telomere maintenance. And in this context, I want to remind you that in 2013, I proposed actually that the methylation age could measure the cumulative effect of an epigenetic maintenance system. In certain ways, it just is a marker of the activity of a maintenance system that maintains the epigenome, broadly defined. Now, these methylation-based biomarkers really relate to a host of age-related conditions. Here I sort the age-related conditions by in alphabetical order. I didn't get a chance to talk about Alzheimer's disease. You notice the middle column also shows you, always reports the source of DNA, because I want to highlight it, that some associations cannot be found in blood. Remember, for example, the post-menopausal treatment in women had a beneficial effect in buccal epithelial cells, but not in blood cells. So you want to be mindful of it. But and also the last column shows you different biomarkers. For example, the multi-tissue, the pheno-H, or the Hannum estimator. And so the biomarkers have slightly different properties. And to highlight that, I want to draw your attention here to a comparative study where I compared the multi-tissue estimator and the Hannum estimator and the pheno-H with respect to different properties. Now, the blue curve shows you the results for the multi-tissue age estimator, and you see, as expected, it works very well at measuring age in different tissues. It relates strongly to centenarian status. It relates to cognitive functioning and so on. But then on other biomarkers, for example, the pheno-H, show a much stronger association with smoking status. For example, the multi-tissue estimator actually does not relate to smoking, at least in blood tissue. It might relate to smoking in other tissues. But by conversely, the pheno-H very much relates to smoking status. And also, on the left-hand side, I show you relationships to leukocyte telomere length. As I mentioned, the pheno-H shows a bit of a correlation, and also the Hannum estimator. By contrast, the multi-tissue estimator is not related to telomere length in a cross-sectional study. And also these biomarkers differ in terms of their predictive accuracy for lifespan. I alluded to it. So for example, the pheno-H outperforms the Hannum, and both outperform the multi-tissue H estimator. So in conclusion, I want to remind you there are several epigenetic clocks. All of them correlate strongly with age, and deviations between the age estimate might reflect genetic reasons, endogenous and exogenous stress, and plain old measurement error. There's no doubt that one could improve the accuracy by coming up with a very careful measurement protocol. Some of these methylation H estimator, certainly the pan tissue, but even more so the skin and blood clock, they do apply to cell culture systems. And maybe they allow us to carry out preclinical studies of anti-aging interventions. I don't believe this methylation H estimator will replace standard clinical measures such as blood pressure, lipid, glucose levels, and so on. They add to them, but will not replace them. Overall, I believe that methylation levels should be measured in many anti-aging clinical trials. In certain ways, if somebody develops a clinical trial and doesn't measure methylation, they kind of need a rationale why they don't measure methylation. So I don't think that methylation will be useful for all anti-aging interventions, but I do believe they would be a very valuable readout for certain and unknown interventions. However, you really want to very carefully think of the source of the DNA. Everybody collects blood, but it could be valuable to collect saliva, buccal cells, urine samples. I didn't get a chance to mention it, but you can very accurately estimate the age of an individual based on DNA extracted from urine. Or some people even collect fat tissue samples. Here's my acknowledgement slide. I'm being supported by the National Institute on Aging and the Paul Allen Frontiers Group and many others. Thank you.
Thank you, Dr. Horvath. The audience has submitted many questions, so let's get to them. Our first question is about your panel and how you identified the various CPG islands that you included in your panel.
Yes, so to develop the epigenetic clock, I used a machine learning algorithm, which is a regression model. More specifically, it's called elastic net regression. And I regressed chronologic age on about 21,000 CPGs that were measured on the Illumina array. And so then the machine learning method picked the 353 markers by carrying out an optimization method. So they were chosen so that they would lead to the best accuracy for predicting chronologic age. I want to mention that all the details of how I developed this algorithm can be found in the supplement of my article from 2013 in Genome Biology, I even include R software code, so it's all out there. And I also want to briefly mention that I'm happy to share all the PowerPoint slides that I presented today. So I can email them to you or distribute it in another way.
Great, thank you very much. Our next question is whether there is a known link between DNA methylation and carrier status for certain genetic diseases. We know you mentioned specifically progeria, Down syndrome, but something like cystic fibrosis where carriers can be generally asymptomatic. Is there a known link between the DNA methylation status and carrier status?
It depends on the disease, you know. So, for example, I don't know the results for cystic fibrosis. I would love to know if somebody should really look at it. For certain diseases, there are surprising results. So, for example, many people know that ApoE4 status is a major risk factor for Alzheimer's disease. And surprisingly, we have not detected a strong, I don't think we have detected a significant association between ApoE4 status and age acceleration, certainly not in blood and not in brain tissue, you know. So some genetic diseases will show an effect and others won't. By contrast, Huntington's disease, which is due to a trinucleotide expansion in the Huntington gene, is actually associated with epigenetic age acceleration both in the brain and in blood tissue.
Very interesting. Thank you very much. I know that you mentioned that there was a correlation between hormone status and the rate of aging, but generally speaking, is epigenetic aging similar across males and females?
No, it turns out that women are slightly younger than men. We looked at it very carefully in blood and also in brain tissue, and it turns out women are maybe 1 or 2 years younger than men when you look at these tissues. So they have a slight benefit. And I also want to briefly mention, we looked also at different ethnic groups, and we published a paper in Genome Biology in 2016 where we showed that Hispanics age slightly more slowly than expected, at least based on the pan-tissue clock. And that could explain this famous Hispanic mortality paradox, where it turns out that Hispanics actually live a few years longer than expected. And this is really a paradox for various reasons. And at least according to the pan-tissue clock, they age more slowly.
Fascinating. Thank you. I just wanted to pass along some kudos. We have several people who are thanking you for this webinar, that they're really enjoying what you're sharing. So thank you for sharing it with us, and we'll move on to our next question. Are you able to explain what leads to an epigenetic mark being durable and heritable versus those that are editable or ephemeral?
No, I don't have a good answer for that. You know, I mean, there are many people who work on that kind of question. There are, of course, quite a bit is known about what kinds of enzymes modify these methyl groups, DNA methyltransferases or TET enzymes and so on. But that's really not my area of expertise.
All right, thank you. This next question, how does methylation age influence the next generation's epigenome? Are slow epigenetic agers more likely to have children who are slow epigenetic agers?
Yes, that's indeed the case. So, for example, we looked at the offspring of centenarians. And it turned out their epigenetic clock actually progressed more slowly compared to age-matched controls. So yes, if you have a parent who is older than 100, then your epigenetic clock probably ticks several years slower than that of a person whose parents have just average life expectancy.
Thank you very much. Do you know if DNA methylation has been studied in the supposedly ageless Hydra vulgaris?
No, I'm not aware of any study in that. I want to mention that many people are interested in studying methylation, of course, in many organisms. Naked mole rat comes to mind, or even bats. For example, a bat can live 30, 40 years, whereas a mouse that has roughly the same size lives only 3 years, And so the question is, what explains the difference in lifespan? And so several people carry out methylation studies then in these different animals to see whether maximum lifespan relates to the ticking rates of these epigenetic clocks. And similarly, people are very interested in the question whether large dogs that tend to live shorter lives than short dogs, whether their methylome differs in some shape or form, So these are all studies that are vigorously pursued by several research groups. Personally, I prefer to focus on vertebrates because vertebrates have cytosine methylation, whereas not all invertebrates have. For example, C. elegans and Drosophila do not have cytosine methylation. They might have another form of methylation, adenine methylation, but just so that you know, there's a fundamental difference between vertebrates and invertebrates when it comes to cytosine methylation.
Thank you. Have you seen any evidence that rate of aging in childhood may be different than rate of aging into adulthood? And might slower methylation age in children be indicative of poor growth?
Yes, I think it's yes to all. So just to echo it. So it turns out that the epigenetic clock definitely ticks faster. There's a much faster rate of change during childhood. If you do the analysis the right way, I need to emphasize, because you need to use the so-called uncalibrated methylation age biomarker. Also, there are now several papers that have looked at how methylation changes according to the epigenetic clock relate to various growth characteristics. And there were some stunning findings that suggest that pubertal speed, for example, the Tanner stage, might relate to epigenetic age acceleration in children. If you send me an email, I point you to some references. I want to emphasize these associations are relatively small. You really need a couple of thousand of individuals to detect it, but there are now several articles that support this view, And it comes back to my comment that the methylation clock does measure development to some extent. And so for example, boys, actually it's a digression, but boys are slightly older than girls at birth. But in general, one would, in children, a higher methylation age is actually desirable. Why? Because it measures development to some extent. It simply indicates the child is ahead of expectation. Of course, later in life, you don't want to have epigenetic age acceleration.
Thank you. Would you please be able to re-explain the concept for TERT alleles being associated with longer telomeres?
Yes, sorry, it is complicated and I rushed it, but yes. So let me start with the finding that certain alleles in the TERT locus are known to be associated with longer telomeres. Why do we know it? Because several large-scale studies have looked at it. And that makes sense, right? Because there will be certain variants that lead to a more active telomerase, and therefore it maintains telomeres better, so people end up with longer telomeres. Now, our finding is that these very same alleles that are associated with longer telomeres, they are actually bad when it comes to the epigenetic readout, because these genetic variants are associated with positive age acceleration according to the epigenetic clock. And that finding is paradoxical because telomere length is a well-established biomarker, obviously, of aging, and one would perhaps expect a consistent association, but it's just not the case, you know.
Thank you very much. We have several questions in light of the fact that you specifically mentioned estrogen and estrogen's potential effect on mitochondria, methylation age. There are several questions about how androgens affect methylation age. Are you able to speak on that?
You know, I really don't know. I'm dying to find out. I would love to collaborate with somebody who could look at that with me, you know. It's obviously it would be super interesting. So one could study really various forms of testosterone treatments, you know, and yeah, I have no data on it.
All right, thank you very much. Our next question, how closely does methylation age correlate with chronological age? Is there always a certain distance from the actual chronological age, or can it vary significantly?
Yeah, well, it depends on the age of the individual, actually. So, for example, today I presented the skin and blood clock, and it's stunningly accurate, especially in young people. If somebody's younger than 20 or younger than 30, in reasonably healthy people, and what I mean by that is in people who don't have leukemia, you know. So in most younger adults, the deviations are, let's say, 2 years or so, 2.5 years at best, and the median error is 2.5 years. And also the correlations are, of course, off the chart high. They are higher than 0.9. However, in older individuals, you can have actually substantial deviation. So you measure the methylation age of a 50-year-old, 60-year-old, 70-year-old, suddenly you can observe deviations of 10, 15 years, you know. And that could be due to various stress factors. We don't quite, as I mentioned several things that are associated with increased aging, for example, obesity, various forms of inflammation, and so on. But yeah, so there you have it. The epigenetic clock is, without a doubt, the most accurate molecular marker of age. You know, so the correlation is in excess of 0.9. Mm-hmm.
Wow, thank you. We know that you mentioned that there are several behavioral lifestyle factors that can influence the rate at which the clock will tick. Have you studied, or has anyone to your knowledge studied the effect of caloric restriction or mimics of caloric restriction on the various epigenetic clocks?
Well, we need to be careful how we define caloric restriction. So when it simply comes to what I call staying lean, you know, avoiding obesity, there's no question that if you avoid obesity, you slow epigenetic aging. Or conversely saying, unfortunately, obese individuals age faster. And that's particularly the case for their liver. The liver tissue ages much faster in obese individuals. Now, but I think the question pertains more to what I would call severe caloric restriction, because some people believe if you almost starve yourself, you're extremely slender to the extent that it's almost dangerous, then the question is whether that could have a beneficial effect. And the answer is we never looked at that certainly not in humans. However, a couple of people have looked at it in the context of mice, where it's well known that caloric restriction, at least during mouse development, has a beneficial effect in most mouse strains. And yes, using then these mouse epigenetic clocks, they have found that caloric restriction is associated with slower epigenetic aging in mice. Yes, I briefly want to mention there are other famous interventions against aging. So for example, there's something called a dwarf mouse that results from knocking out growth receptor hormones, and some publications have shown that these dwarf mice exhibit a slower epigenetic age effect as well. I'll stop with that. Other questions?
All right, great. Yeah. Yes, we actually have time for just one more question, and I think it's a pretty good one. It's very specific, but this researcher is looking for your input. They're looking at DNA methylation age using blood and buccal samples using the same time point in monozygotic twins, but the correlation between them is 0.3. Do you have any explanation or reasons that that correlation may not be as high as would be expected?
Well, the first question is really whether this question pertains to measures of age acceleration or methylation age per se. Because obviously chronologic age confounds the relationship between the methylation age in buccal cells and in blood cells. And if you observe a low correlation, then I usually ask people, what is the age range of your participants? Because imagine all of these twins are aged between 30 and 50, then you would expect a very low correlation. Why? Because not much changes. Whereas if your twins differ in age from, let's say, age 10 to 90, you would expect very strong correlations between the methylation age estimates and the chronologic ages. But coming back to my statement that perhaps the question pertains to these so-called measures of age acceleration, which are really methylation age estimates, but now they have been adjusted for chronologic age. Think of it as the difference between methylation age and chronologic age. And there we do see similar results as reported, meaning that age acceleration in one tissue typically has only a weak correlation with age acceleration in another tissue. And a correlation of 0.3 doesn't surprise me at all. And so why is it so weak? My thinking is that stress factors really have tissue-specific effects. So, for example, certain factors that accelerate the age of blood tissue might not accelerate the age in buccal epithelial cells. Or conversely, it could be that, for example, factors that affect epigenetic aging in buccal cells, for example, hormone levels, don't affect methylation changes in blood. Now, we have done similar studies in other tissues, for example, epigenetic age acceleration in blood versus breast tissue and so on. And that's very much an active area of research, but overall we do see the same results. The correlations tend to be relatively moderate. Yeah.
Thank you very much. Unfortunately, that's all we have time for today. If you have any further questions, and I'm sure you do, as we had over 100 come in during this webinar, please consider reaching directly out to Dr. Horvath. His email address is shown on the screen. As a reminder, the webinar will be archived on the scientist's website, and you'll receive an email notifying you when the on-demand webinar is available. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your great questions and comments. On behalf of the scientist, I'd also like to thank our speaker, Dr. Horvath, as well as our webinar sponsors, Zymo, Active Motif, and Life Epigenetics. Thanks, everyone, and goodbye.
Thank you.